since its formation of ADCOM, and he was the General Secretary of IPA, AP State Branch for 3 terms, and Founder Secretary of Treasury and IPA Regulatory Affairs Divisions for 3 terms. He served as Secretary LOC of the Indian Pharmaceutical Congress 2000 in Hyderabad, and he was a active core committee member of LOC of 66th IPC in Hyderabad that was successfully organized by the All India Drugs Control Business Confederation. He was one of the mentor of that conference. Dr. Bir Raju filed and got approval from US FDA a couple of dozens of ANDAs and DMFs, sterile dosage forms, transdermal patches, topical dosage form, aerosols, sprays, nasal delivery system, and oral solids and liquids. Dr. Bir Raju successfully completed many US FDA audits. He traveled across the globe for vendors, CMOs, and CROs. So, with these few words, I welcome once again Dr. Bir Raju and request him to give his introductory remarks about the topic. Thank you.
Thank you, Dr. Jayanta, for your kind words. So, welcome, Dr. Satya Boturi. So, today's topic is very, very apt and important for all our, regulatory fraternity, the drugs control fraternity, to understand the implications of monitoring and non-monitoring the different types of impurities we found in pharmaceuticals. The origin could be different, their method of generation could be different, but these impurities need to be monitored. These need to be quantified, need to be controlled. So today's topic is very, very apt, because most of the times, the people ask, they consume all sorts of irrelevant things like, roadside charts, roadside food, the contaminated water. So all sorts of things they consume, but, they go to a doctor and ask for, do the medicine has a side effect? So that's such a situation we are in. So the importance of this impurity monitoring, so that it doesn't add to the misery of the suffering. So that this could be one less headache if these impurities are controlled in pharmaceuticals. And there are, I think most of you know, there are certain impurities which can trigger a carcinogenic alert. So these are found in minute micrograms or picogram level even. Even then, the FDA, the requirements internationally is to control them, quantify them, and limit their presence in all sorts of pharmaceuticals like APIs, drug products, etc. So the impurities, one of the FDA's refuse to receiver criteria is, if you don't limit your unknown impurity to a certain level, FDA will refuse to accept the application even for review. This is such an important thing. The unknown impurity, maximum single largest unknown impurity quantification, is not properly justified. So the application, even FDA will refuse to receive and accept for review. So this is such an important topic, and there are advanced tools available. There are different sources from where the impurities generate. Like Dr. Jayanta mentioned, it could be an interaction between the excipient and the active. It could be due to the environmental factors, or it could be due to the degradation of API itself. So these impurities needs to be characterized, needs to be identified, needs to be quantified, and needs to be qualified. I think we are fortunate to have Dr. Satya here, with his vast experience. He will walk us through important aspects of all these, impurity, from generation to quantification to control. So I feel it, very apt and interesting, and I feel proud to be part of this today's 31st seminar organized by our All India Drugs Control Officers Confederation. So let's enjoy, and I welcome Dr. Satya. Now, please go ahead with your enlightenment to all our fraternity. Thank you.
Wonderful, sir.
Thank you.
Good morning to all of you. And I'm sorry, go ahead, Jayanta sir.
Thank you, Dr. V. Raju, sir. Dr. Raju is joining with us right from the US. So we are glad to hear your voice clearly and from the US. And sir, thank you for your encouraging words and introductory remarks. So let us hear the more insights about the topic. So as per protocol, it's my duty to introduce Dr. Satya Mathavidi before the audience to brief about the profile of Dr. Satya Mathavidi. Uh, Dr. Satya Mathavidi is received PhD from JNT University, Hyderabad in pharmaceutical chemistry and MBA in international business from Andhra University. Dr. Mathavidi spent about 27 years in various pharmaceutical industries such as Dr. Reddy's, Zydus, Sai Life Sciences, Granules India, and Biological Events Limited. He has huge number of successful regulatory inspection to his account and has been recognized as a subject matter expert for pharmaceutical quality and regulatory affairs, managing the multi-site pharmaceutical operations. His expertise include quality and manufacturing operations, regulatory and scientific affairs of INDs, ANDAs, and DMF for all kinds of pharmaceuticals, including APIs, drug products, oral solids, injectables, long-acting injectables, complex generics, and peptides. Dr. Satya Mathavidi is a proven leader and mentor for complete value chain of pharmaceuticals, including product development, operations, quality, and regulatory and scientific affairs. He is currently with Biological Event Limited as a Senior Vice President, Head of Operations, Quality, and Scientific Affairs. So I am so proud to have with us Dr. Satya Mathavidi. Now I am hand over the stage to Dr. Mathavidi for his insightful presentation of today's topic.
Thank you. Thank you, Dr. Jayanta, sir. I think to be here with all this seniors, and the mentor for the veterans in the regulatory industry, and also the regulators from the Indian Drug Regulatory Authority. Am I audible to all?
Yes, yes.
Wonderful. Yes.
Wonderful. So to be here in this forum. And to be honest, this is actually after a long gap. I'm back here onto this forum. I used to participate earlier in offline programs, one program in West Bengal and a couple of programs in Hyderabad, and there are some other events. This is a wonderful process or a program that is initiation from the EDCOC, where this is in a need of the hour. I think we need a constant, push in being up to date with the market and the technological advancements. So being in the past 27 years, I try to justify the opportunity that is given to if there are the seniors over here or the mentors over here. As you all know, the subject of impurities in pharmaceuticals itself is a vast subject. Simple, you need to understand from various and the level and understanding or depth of subject is actually commensurate that you working with the generics or the new drug entities or NDCs or NDEs, what we talk about, new chemical moieties. So the right now the current talk I would like to limit to a moderate, try to understand to some extent the basics, most importantly on the sources of these impurities, how the reacting to this, how regulators are reacting it, and how the regulatory focus is actually shaping up across the globe as far as generic pharmaceutical industry is concerned. I'm trying to keep a low light on the new drugs because that itself is a different subject where you need to understand more about the research associated, the characterizations, safety studies or clinical studies. So I'm keeping that part away from this discussion today because I want to focus more on the generics because the generics is actually more prevalent the growing industry as far as India is concerned, and we is where world is going and where we ourselves as an Indians stand in this whole race. I think that makes sense for the uh, let's back to the subject. The subject for the today is the impurities changing historically. When I start my career in 1990s, the way we look at the impurity controls, impurity profile, the regulatory submissions of DMFs or the ANDAs was completely different what we are doing it today. Though there are regional differences, there is a significant focus or a shift by the in terms of the impurities control in your pharmaceutical products in any form. It could be any kind of dosage form, whether it is solid oral, liquid oral, injectable, or any other topical product. I'll try to touch upon the basics of impurities. I spend more time on these because you're all well versed with these kind of things. You're practicing regulators for this control of impurities and within your own regions. I just wanted to touch the basis so that we can set the next level of discussion, which is actually sources. How do you control what is expected from industry, what is expected from the regulators, and what kind of advantage we are offering to the patients at the end of the day. That's going to be the major focus of my discussion today. And I'm also try to pull up some citations from the major regulators in the review of the applications, whether it is DMF or in CTD format or ANDA or INDs or any other kind of laboratory assessments in terms during the process of drug approval. I think, are there any other expectations which we should cover in this program today so that I can touch upon? This is the basic idea I have for the day.
Yeah, that's such a you're apt on if you focus more on the generics, because in India we have mostly the generic products and generic approvals. So if you limit the talk and more focus on the generic aspect, it's I think very
let's quickly back onto the subject. So as you know, anything other than the therapeutically active moiety introducing into the body, for me, it's an impurity. Impurity is not necessarily be an organic product, or it can be an inorganic, it can be a physical, it can be any kind of other foreign matter that carries through the either the therapeutic moiety or through the vehicle which you are using. I mean, vehicle in the sense the excipients. So anything which is other than the therapeutically active moiety is an impurity for me, which are supposed to be controlled to the safest level based on the data that is produced. Since we are talking about a generic industry or a generic drugs, always we have a standard in place or a drug in place, which is already proven to be safe for the human being at a. That means the profile of impurity that is, it is inorganic, organic, or any other physical impurities or a microbial are actually known, at least at this point of time in a generic industry. So as a manufacturer of a generic drug, the first liability we have, first responsibility we have is to match with the reference listed drug, which is already approved either in India or somewhere else, so that we can take the reference of the same that is generated as a part of the drug development or approval process. So those impurities you need to match with organic impurities, and especially this is also many times are ignored, and we are focused onto the chemical impurities or the synthetic impurities, and in characterizing them. Some extent beyond the level, the few salts, the inorganic salts, and some chemical reagents we use, and be a filtrate or some metals, for example, could be also be a threatening the safety of the product that we administer. And much gone ahead in terms of the impurities with respect to impurities or residual solvents or mutagenic impurities, especially. Recent focus or the major regulators have a basic on these impurities or assessment of mutagenic impurities as part of the approval process. So the other kind of impurities could be, Dr. Raju sir said, there could be, the regenerative or a co-reactive degradation products or a compatibility related products, the kind of interaction with the drug packaging material, the environmental conditions. So these kind of impurities also must be taken into account before you actually go for a submission for for approval to the authorities. When we started our career in 96 with my first drug master file of ibuprofen in Dr. Reddy's, the way on the impurity profiling as a regulatory affairs specialist that time versus what we are doing today as a part of our A&E assessment is totally different. We have came a long way, but in my opinion, that's still not enough. Why I say it's still not enough, probably I'll cover it in the further slides going forward. Now, this is an interesting factor or a kind of subject for me. The sources of impurities partners, be in active ingredient manufacturers or excipients or a primary packaging manufacturers. Most of the times, the is not at the desired level. Even when we talk about the few regulators, they are more or less reliant on the pharma. Since you have a monograph, I'm okay with the monograph, but monographs are, as you know, were developed long back, going back to the 1948, when the Indian Pharmacopoeia Commission was set up. Hope I'm right with that year. So in 1948, when the Indian Pharmacopoeia Congress is set up and the first draft is released, and after that, the few revisions, most of them were actually copied from the British Pharmacopoeia or later on European Pharmacopoeia, and they are based on the technology that is exist that time. For example, you all agree with me. We used to work on impurity control on the TLC for many of the products, the thin layer chromatography, where it is not very sensitive, not very robust, not very reproducible. And we largely depend upon the spot we see, the absorption, which is a kind of a semi quantitative method. From that point, we traveled a lot, talking about LC-MS-MS, we talking about LC quadrupole, coupled with the mass spectra, which are the and other things, and we are able to characterize and identify at a ppb level. That's where actually we traveled. So that there is a lot of, and there is a need now to revisit most of these products and see the level of or the appropriate level control that product. I know it takes its own time to write this monograph, the consultation part, and other the data generation, data collection, review. But I think there is a need to relook control of these impurities in the products we consume, at least in our own country, and the relatives and the family. Now, sources, I would, this I even as a layman, easy to understand from where these impurities can be introduced. These impurities can be introduced from the inputs I'm taking, whether it is API or intermediate or packaging material or any other chemical which I'm using it. Those are all called inputs. And the ancillaries of the production, for example, if I'm using a filters, if I'm using a kind of a silica gel for acid filtration, if I'm using some kind of tubing or hose pipes which are made of different kind of polymers, these are all maybe chemically active, chemically inactive. We don't know unless we actually focus onto this. As you all know, most of the synthetic products, synthetic drugs are chemically derived. I mean, when I said chemically derived, they are through chemical reactions. So when you talk about A plus B equal to AB, it can also be A1 and B1 that are added as a part of the manufacturing process, or yes, surfaces that touch during the reaction mechanism. That the part of the understanding of the process development also much in the, at least in the Indian industry and of course across the globe, that the compatibility studies, we call it as technically coupon studies in our product development strategy, is also now taking care of the MOCs, the material of construction of various surfaces that are to have to touch during this processing were also studied and see impurities that can leach or that can generate during this chemical reaction processes. So that's about the impurities from the the impurities from the input from this compatibility point of view. And as you all know, the any synthesis of a drug product involves many steps of reactions. So there could be 1, 2, at the max. What I have seen in my career so far is 72 steps of synthesis, which will yield your desired activity. So the control of your intermediates, whether it is isolated or not isolated, at each step, control of quality of impurity, quality of the drug, the API at each step or intermediate step is very, very essential. And at least the knowledge of the impurity formation, impurity carryover at the various steps, and finally landing into the API. This knowledge and understanding is as a part of the process development. Otherwise, you will end up with the surprises in the API from where this impurity is coming up. Because main reaction, always there will be a co-reactions or a parallel reactions that could along with the impurity moiety as such. So it depends on the exist as an impurity within the active moiety and how that are favoring for the or they are catalyzing the chemical reactions. So we must aware of the synthetic intermediates quality, impurity purging, and also the byproducts, potential byproducts that have potential to carry to the final product. So this is very much known, the chemistry related, of course, which we talked about right now. Though there are no chemistry involved in the formulation process, basically a mechanical or the physical process of making the drug solid oral, liquid oral, or injectable, there could be a potential impurities as well, because again it comes with the interactions between the inactive ingredients used as a vehicle and the material or MOC compatibility of the equipment train and the kind of use during the formulation process can add up these impurities. If we take an example of a formulation drug product, for example, though it looks very simple, it just you need to note and make it into a solution form, any of the vehicle, water or any other form, and then you fill it. So that's the basic process, aseptic fill or a terminally sterilized fill. But there are a lot of ancillaries in process, right from dissolution of API into your vehicle, followed by the filtrations. So whether your filters are really compatible with the drug product option, is there any kind of leaching that happens, is there any other impurities that can ingress into the product in connection with the time, because everything is important. Your time, the whole time is important, your exposure time is important, or any other environmental conditions that can affect your product in a solution form. The overall filling time, the potential for ingression of any during this process. So how good your aseptic processes are, how your aseptic the systems and the behavior is, your operators behave, and how well you control with other primary packaging material for your drug product, either it should be a rubber bung or a vial or ampule or any other. So these are all more important to understand where an ingression of these impurities to your product. You cannot say that my API is controlled for these 3 impurities. Pharmacopoeial monographies has only mentioned about these 3 impurities. I'll end up with these 3. That may not be sufficient nowadays. Again, it's a region specific. I have seen a huge variety of differences between the various countries across the globe, starting from India to rest of the world, to the most developed countries or a highly regulated countries like US, Europe, and Japan. Apart from this, there are on not just organic impurities. You must also understand the isomers that can be associated, especially when there is the chiral carbons product. So you need to understand the therapeutic of these isomers. Not necessarily that all isomers will have the same kind of therapeutic activity, though the structure is same. Only the spatial arrangement is different. But the many cases, fatal cases that are reported across the globe, where different isomers have a different kind of effect on the human physiology. And often this is again, there was actually a period of time. We were also now considerate about the morphic impurities, polymorphic impurities. The structure is same, the active chemical moiety is same, but is a different kind of the structural form or a crystal lattice we talk about. Then that also nowadays is considered as an impurity. Not necessarily that it will have an undesirable effect for the human physiology, but it can have certainly in terms of the solid oral dosage forms, as it can affect your the ADE of your drug product, which will be comparable with the innovator drug product or a reference listed drug product, is RLD. So the polymorphic form impurity is also important to understand. We call it as a crystallization related impurities. And not solvents, of course, are also important to go to control within the guidelines that are published internationally, ICH basically, and also the other regulators. Often it is ignored. We also talk about the recovered solvents. There are many cases where the impurities were actually got drug product and there were recalls from the market, especially in the regulated markets. But when actually gone back to the investigation, to their surprise, there are plenty of cases where the impurities were introduced from a recovered solvents. So that could be one of the source, which is basically going back to our first input related impurities. You also must control your apart from these. So I think this is where should address that. How do we control all these kind of impurities within the level of within the safest level when you administer the dose? As you all know, any even for me as a drug product which you are administering as a chemical or whatever it is, that is foreign body to the foreign body for the human physiology. So adaptability is how the ADE administration takes place in the human physiology is also important, and we need to match those parameters when we talk about the generics. Otherwise, it has no meaning, because we are not repeating all the basic studies to understand the safety level of this drug as a generic manufacturers. Definition itself, we are reliant on the safety data that is or published by the innovator as a part of the drug development and approval. We are not repeating those. We are largely dependent on all the clinical data that is. That means we have an essential responsibility on all of us, industry, and of course, as an approving body regulators, that we align, we comply with the levels of these contaminants product at par or less than what is proved in the RLD. Obviously, we are supposed to, as a first step, we are supposed to RLD. But the whole subject is actually complex, because there could be different routes of synthesis for API. Not necessarily I will be following the innovator route of synthesis for my chemical synthesis API. Not necessarily I will be following the same equipment train, same kind of the exposures or a product exposure when the innovator is formulated the drug product. So I develop my own formulation process, will be complying to the Q1 and Q2 of the innovator. So that means the basic things we are matching, but many of the variables are actually not in control. And either it is because of the data that is unavailable from the innovator, or that's not published from the innovator, and many of these are subject to change. So you cannot have, I cannot insist as a regulator, for example, to have the similar process, similar formulation process, similar API process, similar test methods for of products that are being marketed as a generics. That's a basic challenge for the regulators as well, because unless otherwise you match exactly with the innovator, and also you have robust processes in-house. If you don't have a robust process in-house for the impurity profiling, the complete impurity profiling, it will be difficult to achieve drug product. As you know, and it is not actually easy to standardize also. When we talk about impurity profiling, it is actually different from the impurities. The impurities, the individual impurities, as we discussed in the beginning, the various kinds of impurities. But study that encompasses, that enlists all of impurities, all kind of potential impurities, whatever we discussed in the first slide. And you, or as a manufacturer, will conduct a thorough review on those, talking about the risk assessment or a gap assessment. This quality risk assessment has to involve all kind of inputs you take, all kind of chemical process that takes place during the processing, and all kinds of the compatibility issues with the equipment, with the environment, and also the condition about the storage, primary packaging materials, and the ability of the test methods to detect if anything goes wrong. If unless otherwise I do this study, I cannot assure the drug product. As I saying, if I'm complying to the monograph specs, have a limit for impurities. Most of our monographs, unfortunately, have a limit for unknown impurity by TLC or by HPLC. Some new monographs. Most of them are age-old monographs. So is that enough? Definitely not enough. And not necessarily that we should apply the compendial specs or binding. Of course, they are legally binding a lot beyond those published monographs. When as a quality assurance person, I insist all my that do a robust impurity profiling, considering all these before you go on to a scale up, so that we will have a fair idea, so that and involve the process engineering team to design a suitable process for this when we go for a commercial scale. That's how we need to actually have a control in place. And beyond that, we should also focus more onto the analytical resources to have a robust and rugged method in place, which can tell us if there is any the riders with respect to the stability indicativeness of the product. My product is able to detect a particular impurity, for which anyway we touch this subject in the further slides, for which we need to see the product at extreme and see how it behaves, whether my methods are able to that out and then give you an alarm that so and so impurities or so and so impurities are popping up at so and so conditions, so that I would be able to design my store conditions, my packaging conditions accordingly. So as far as the control strategies, whether it is explicitly stated in the guidelines or not, because as I said, regional specifics, the primary onus is on the manufacturer as the owner of the product, as the developer of the product, to understand and carefully select the raw materials. As you know, one compound, one chemical, many synthetic ways. You can start from X, you can start from Z, and end up with the product that you need. So each chemical, it has its own advantages and disadvantages. So at the industry, what we see is my process must be cost effective, will be less in in the time cycle, or easy to handle. No hazardous chemicals involved. No no environmentally no damaging things that are happening. If the effluent is in good control, or the availability of those chemicals or freely available in the free trade of commerce. So all these kind of we take into account when we select materials and the route of synthesis. Not just, say for example, if it is so cost effective but it is environmentally detrimental, we may not go for it. If it is so cost effective but there are hazardous materials involved which will be a threat to the operators or safety of the operators, then we may not go for it. So we do all this, the risk assessment, when we select a final process to get into the API to synthesize the API. So there should be an appropriate control and due diligence over selection of starting materials and their control of impurities at that level. I think you are all aware, we are also nowadays the industry budgets about the QBD. When we were not used to talk about the QBD, with the overall drug product quality controlled through specifications. It's called control by testing. Now we are talking about a different subject, control by design, quality by design. So as you all are aware, so you cannot test all your millions of drug product for the quality, right? It's a random check. You select your units from your drug product randomly and then check for the quality. And those results are interpreted across your batch and all units of your batch. That means if your sampling approach is not scientifically or statistically valid, and your the ancillaries or other things during the processing controlled, there could be every chance of other impurities which can go undetected and get onto the human body as an impurity. So we must be very, very aware of all these, how the selection of starting materials and how quality of your starting materials are controlled, how compatibility issues are understood during the entire manufacturing process development. That's we call it as an ideologist. And as to what extent whether the industry is really practicing this, it is up to the industry and the regulators. But end of the day, that's the need of the hour. And many of the industries actually do this because from the person from the industry, we understand the cost of poor quality, the cost of the poor OTIF, on time and full, the cost of poor first time. We all understand this cost of compliance actions or regulatory actions, shutting down the facility or recall or kind of the seizures or any other the penalties for the company's credibility, and also the will be very detrimental. We understand. I think the industry understand that very well. To the maximum extent, all these things are taken care, at least by a moderator or the cream of industries within India. And there are a lot of guidelines available how to control the impurities nowadays. There is a very less room for the at least now. Earlier there are no such guidelines. We don't know how to set the entire impurity profile or how to design the entire impurity profiling protocol. Impurities, how to set the specifications for the various kinds of materials within the process. But now if you ICHQ 6A, where it's very, very clear about the strategies, option 1, option 2, option 3 for setting of acceptable impurities or specifications for impurities at various specs, various stages. It is also important because these are chemically active or reactive, and as a foreign body within the or a foreign analyte within this within the drug product, we must also understand what kind of and when they are actually introduced, whether they are introduced during the of the process or during the transfer or during the storage or during the environmental conditions or during the logistics, I mean transportation, we don't know. So these things understood clearly and set the limits and the control strategies for those. And there are many instances where we also talk about the purging of impurities during the process from step A to step C, for example. If there are a couple of impurities that are during or generated during step A, we will see with the same kind of to what extent those were present in step 2, whether they're purged, or is there any need for the washing of that particular one, or eliminating that impurity by some other means in the processing itself. And of late, the industry has also realized that the process engineering would also be helpful in managing life cycle. It is not just enough the chemistry. Chemistry is only for the design, but it's more or less the mechanics, the more or less the setup or engineering setup that can actually standardize the manufacturing processes. ppm selection, I think it is also important to control the impurities. So though there are some countries who are not actually asking for the extractables and leachables data, only they are relying on the food grade certificate, but that's not enough for assuring the quality or assuring the impurities ingression or no ingression from these materials is also need to be proven. For example, the product which we call it as the extractable and leachable from this product, from these primary packaging materials, whether it is the plastic, whether it is HDPE, LDPE, or your glass vials, ampules, or a plastic vials. So these need to be understood in detail. There are a lot of domain for various strategies how to study the extractables and leachables, kind of testing you need to employ, and how to interpret those extractables, leachable impurities to the safest level. And finally, if all these do not for the industry, and of course, if also for the regulators, if they are not acceptable, still up with some unknown impurities beyond the levels that are published, or beyond the levels that are believed to be safe, need to produce, need to go for further safety studies, either using MS test for 90 days or 60 days, depends on the kind of impurity we talk about, and justify the level of the impurities at the final product nearly versus the treatment period, the administration period, or the overall exposure, the maximum daily dose plus the period of actually justify the level of impurity that has potential to present finished product. So this is about there are various control strategies available for the industry at this point of time with all the known knowledge of the generic industry. Any questions maybe you can ask in between, or we can reserve for the end. Hope that should be okay.
Yeah, we'll take them at the end. Yeah, go ahead.
Right. So that's about the control strategies that are available, options available for the industry. That's what we discussed in the previous slide. And I'm trying to put up more of my experience, which I learned dealing with the many regulators across the globe, including US, countries of Europe, and again in Europe, Central Europe, Western Europe, and the Eastern Europe, and the BRIC countries, Asian countries, or a kind of Latin. There are different kind of experience, and sometimes it will be actually surprising to see, but surprising to see the expectations from these countries, and some to go through, and some countries they have the unexpected questions or surprise questions. So these are all part of our day. I would like to dwell some time here in looking at the regional differences, since we should talk about our home first. The controls over impurities in Indian scenario as of now, in the beginning, we are mostly reliant on the monograph controls. If the monograph product is passing after the monograph, you are done. But I could see there are a lot of the insights by the regulatory audit, the Indian Drug Control Authority in the recent audits which we faced, or with your drug inspectors, was really encouraging. I'm really happy to see that they were talking now about the impurity profile, about the specific specified impurities. They're talking about the known unknown impurities, purging studies, of residual solvents, and they are talking about the validity of your studies or degradation studies to see the potential contaminants or degradation path and the shelf life quality or the quality at the shelf. I think that we are on the right track, certainly, but there is a improvement. I hope the regulators are taking care of this. When we talk about the regional differences, some countries are based on their own the guidelines. Especially we talk about the rest of the markets. They have their own set of guidelines. Some are very intensive, go through, and some are very critical or very cumbersome process. And for example, when we are working for a product called a product which is with an antithrombotic or product, which is enoxaparin drug product, the control, the regulatory control exercised by the various regions were totally different. For example, the US for approval of that product versus the way we deal with the European countries is totally different. The expectations are different, regulations are different, and the kind of studies they need are all different. Probably in future, we expect that there will be an harmonization across the major regulators, so that for the industry, it will be easy for us to set a uniform standard of a drug product across the globe. Expectations from at least 1996, I can relate now. From 2022, the regulatory expectations are the way gone up, and we are now talking about embracing the new technologies, analytical technologies that are available to control these impurities to the maximum possible extent. And we also talk about the quality risk assessment. We also talk about impurities and the process efficiency, so that you can actually limit those impurities to the desired level. And I hope you all agree with me. There are the certain concepts that are coming in of the continuous process improvement or a called optimization during the product life cycle. Is the concepts also are taking up now, wherein if you're controlling the impurity to the limit, it's not just enough. You may have to tighten, or you may have to tune the product to reduce the impurity to 30% of the spec level. So those are the expectations now coming up from the Western. I think they're good for the company, but good for the patient. Also involves the expense and also time for the development. The way now the regulators are looking at the overall controls, they expect a robust impurity profile process as a part of your process development. Especially in generics, as you said, there are many control strategies available, which is ancillaries or control of your the components or chemical reaction processes, control through various other mechanisms, especially protecting the product from the potential damage from the environmental conditions, light, water, or any other heat or any other conditions that can damage your. But now it is not enough. Now they're asking for a proper impurity profile, all kinds of sources of impurities and all kinds of impurities that have a potential, which again essentially go back to my first and second slides. All those must be listed, and we need to explain why and how impurities are controlled in my drug product or in my drug substance. So there are discuss in the next slide. Is my experience also from the questions from the regulators as a part of the approval process. They are very much keen. The regulators are very much keen now about the thresholds of the impurities. So what could be your reporting threshold of, for example, if the dose is less than 2 grams per day, what should be your reporting threshold? 0.05% by HPLC should be your reporting threshold. Any impurity peak or any impurity that is more than 0.05, you're supposed to report part of your COA. And similarly, the identification threshold is also clear now. So if any impurity more than 0.1%, you are supposed to identify what is that impurity. Identify in the sense you may have to characterize the structure, you may have to develop an appropriate analytical method, validate the analytical method, and see whether that impurity is being is being identified and quantified with reasonable precision and accuracy. And any impurity, any unknown impurity, any impurity beyond 0.15 is now called as a qualification threshold. It means you need to justify why the 0.15% of this is acceptable, whether it is the data available from the RLD or the data available from clinical studies, or the manufacturer themselves has generated any by some kind of qualification studies or in vivo clinical studies. And the thrust degradation studies was also more nowadays from the regulators, because that's the only way to see the extremes of your product behavior. If I store my product at the prescribed storage conditions, it may or may not give overall picture of the extreme behavior of my product. So there are pretty much, the guidelines are very much clear now. They are available across the globe, that what kind of the extreme conditions we need to see and study your product, expose your product and see the behavior are very much clear now. For example, degradation, base degradation, water degradations, or kind of oxidations, light degradations, all these must be studied, because that will give you an understanding of the product behavior at extreme conditions. Right from the release of the material, release of the drug product from the manufacturer's facility to reaching the patient shelf. So anything can happen. Exposed, it can be exposed to light, it can be exposed to the harsh environmental conditions, light, or it could be temperature, could be humidity, or could be any other. So see the product adaptability of all these conditions and see if anything can actually create. The thrust on these degradation studies has really got a lot many questions when the reviewers review the application for. We have a few things listed in the second slide. And suitability, sensitivity of the method. This is another area mostly ignored. Many of the countries or many of the regulators as a part of the approval. The detectability or the estimation or determination of impurity through a good analytical method. Even if I derive 10 synthetic pathways or a degradation pathways or impurity pathways out of my research, if I don't have analytical support or the advanced, I cannot be able to properly isolate, quantify, and characterize those impurities so that control for my release, for my finished product release. So the development of a process, the development of the methods, the development of the drug has to go hand in hand with the analytical research team as well. If you will land up with the false positives, or sometimes you'll not be able to identify the contamination that has happened to the product. There are a lot many questions, or there is a lot of advanced characterization of impurities, whether it is known or unknown, specified or unspecified. There is a characterization has a major thrust nowadays. Regulators were asking about how do you characterize the structure and then release this as a standard for the regular control in your product. And there's a due consideration to all other impurities, not just organic as earlier. Unlike earlier talking about the organic impurities, now there are a lot of questions about the other inorganic impurities, the particulate impurities, be physical impurities within the product or any other things. And more the mutagenic impurities. So mutagenic impurities, as you know, any impurity reactive mechanism with the DNA can be called as a mutagenic, also be called carcinogen or any of the other names, but it is prevalently be known as the mutagenic impurities. That means which can alter your gene, which your DNA, and then can cause to many fatal, life-threatening things as a result. So now, how do we control those? Any chemical by nature could be a carcinogen. We don't know any chemical because there won't be any compatibility as the human cells. So now, since the chemicals are only our inputs, especially in the synthetic drug industry, we must be very, very careful in the cell process and their interaction with the DNA. Of methods that are already published, I think the world has gone way ahead in terms of assessment, mutagenic assessment of the drug product, the kind of compound by high reactivity with the DNA. For example, nitrosamines, we talked about any amino alkyl products, and primary amines, tertiary amines. So these kind of products are believed to have a high reactivity with the DNA, so that they could be limited. And anything you be using those, or you may not be using the products with those impurities as input, but there would be every possibility as a part of a byproduct or as a part of the co-reaction that can happen, and generate these genotoxic impurities. As an industry and as a regulator, you must also look at the potential for genotoxicities based on the route of synthesis, which we follow, based on the equipment train and operational controls we have. We must see potential for these impurities and ask for the genotoxic impurity assessment. There are certain limits, general limits that are applied, but again, it is largely due on the risk assessment and the data that we have either in the public domain or with the manufacturer before we justify the mutagenic impurities. The TT total toxicological concern of impurity is 1.5 per day, though they were given that as a general criteria, but in my opinion, that's not just enough. You may have to have a protocol for the genotoxic assessment or any structural alerts that you have in the process or in the product, which could have a potential genotoxicity verified and justified through the data, not just through the beliefs. Times I could see the industry says, because we also deal with many of our API partners, across the globe. We get some statements saying that this identification of this impurity is impossible. So there is nothing like impossible, and this is not accepted, as a consumer, and also as a regulator. So difficult to synthesize, difficult to identify, or difficult to assess are not the acceptable terms anymore. That's the expectations that was actually evolved, or the regulatory expectation that was evolved over the years. These are the few citations I collected out also from the public domain, and these are the queries what we receive from the reviewers of our application, either as an API manufacturer or as a drug product manufacturer, which actually gives you an indirect understanding of how the regulators were actually evolving with this subject and how critical they are in assessing the impurity profile of the product beyond the legal expectations, which is monograph. There are citations, there are plenty of questions from the reviewers that the impurity is insufficient. Like, for example, I only have a three which based on which I may be proposing a limit, may not be enough. Nowadays, we are talking about design of experiments, quality by design. We are talking about a statistic acceptable limits, that the statistical data. Unless otherwise we have a sufficient data from the process development exercise, this is very, very difficult to understand the nature and the profile of impurity so that we can effectively control in our drug product. And there are, in my many questions about the process efficiency. For removal of those impurities or control of up to 0.8, not 0.08 or 0.9, which were very close to the spec. Technically, they are passing, but I would say the process is not in control. How do you? It can be because there could be an inherent analytical error as well, depending on the technique you use. If you are using an azole technique of a semi-quantitative methods, there could be more than 0.08 versus 0.1. It's not passing. It's failing indeed. It's beyond 0.1 as well. Even if you see the well-evolved technique of H, there could be an inherent error of 2%. So either it could be 0.09 or it could be 1.1. We don't know. So that's how actually we need to evaluate. Even as a regulators, we need to see the efficiency or the inherent variation in the analytical methods that are applied for control of these impurities. And also the process efficiency versus this behavior near to the expiry is also important to understand to control. Statement, no historical data on impurity profiling. This was very seen predominantly in many of the API processes or the DMFs as a part of the DMF review. So probably some manufacturers may the validation of the process, validation of the method in the beginning, but over a period of time they may not be back accounting all the minor changes they made during the process over a period of time, and whether the process or the method is performing the same way what is actually validated. They don't see the or a requalification of those processes, and that is one of the reviewer points seen in many cases. And this is interesting. So when we especially the API partners across the globe, predominantly from Asia and the LATAM, most of the times we get an answer saying that identify. So when we talk about, okay, what is this unknown impurity which is appearing at this stage and this there in the so many batches, what is that? So they say it's difficult to identify. It is 0.05, for example, less than 0.1. That may not be enough sometimes, because until unless you have a genotoxic impurity assessment, I cannot believe that this 0.09 impurity, which is less than identification threshold as a chemical impurity, whether it is having effect or no. So that's how we need to understand all the within the drug product and relate something and then decide whether it. And there are a lot of questions where about impurities are not adequately identified, qualified, or characterized. So sometimes also have a difficulty in managing our vendors, saying that, okay, I have identification by the area percent or the peak or by retention time. If I don't know if we don't have the standard, it's not properly proven to be the response factor is not properly proven, and if that is not properly controlled by method means difficult for me to rely just alone on the retention time. So the retention time could vary. The two different compounds can get at the same retention time. We don't know. So in that case, as a regulator and as a consumer like us, I'm talking about as a finished product manufacturer, as a regulator, it's also important to see the rationale, whether how this impurity is being controlled, whether your method is really capable of resolving this impurity, capable of identifying with acceptable recovery and resolution. This is most important to see, whether this impurity is being really controlled. Otherwise, we will end up with the difficulty in getting the approvals. As industry, of course, as a regulator, you have a challenge in getting all this data. Impurities are not adequately identified, qualified, or characterized, more or less same as we discussed earlier. And if you're not, the trend data that you get from the process, because we actually assessment or quality management principles where we need to assess the process capability or a process efficiency at least annually to see the quality parameters of the product, critical quality attributes of the product are behaving, so that I can see if there is any switch, if there's any that can actually trigger from this data. If the data is not to the trend, certainly as a regulator, we should also focus on these alarms and see the potential impact that can happen through some kind of a predictive analysis. Unknown impurities are in product are alarmingly high. They may ask you for the data, and polymorphic impurities are not properly controlled. So especially the different polymers can have a different deletion rates, so which can directly impact the bioavailability of actually in the solid orals or liquid orals, where it can differ the dissolution profile that has been established as a part of RLD versus the generic product. If that is not, if the dissolution profile bioavailability is not same as RLD, then the generic should not be approved. The understanding between the industry and of course the expectations from the regulators. I are not properly controlled, and degradation studies were not sufficient. For example, the whole exercise, the whole intention of doing this stress studies is to understand the extremes of the behavior at the extreme conditions, and more importantly, the reliability of the analytical method, the capability of analytical method to isolate, detect those changes in the product with these extreme conditions. This as a stability indicating method. That means my method is able to detect the changes that happens during the stability of the drug product. Affected by many factors, can be affected by the storage time, can be affected by storage conditions, can be affected by the exposure to the various environmental conditions, extreme conditions. So these are studied, and the data from these studies is very, very important to understand or justify the storage period versus the storage or the primary packaging materials versus the conditions that you should live. So, and often this is actually ignored. Most of the companies, they do a very minimal study, part of their method validations, but I would advise this is a very important document which gives you an insight of the complete profile of the product or a stability profile of the product. And there are some questions, which we could see from the regulators about the control of these impurities from inputs, especially the starting materials, key starting materials, and byproducts, and the issues in the characterization. Just like I have seen some cases where we just run an infrared spectroscopy, IR spectroscopy or UV, or coupled with HPLC, and say that this is the impurity. So with that, we cannot really derive the structure of that impurity. We must see the proton NMR, carbon NMR coupled with the mass spectrum, at least, if not the isomers like the COSY and all. At least you should have a sufficient data to derive the structure of this impurity, so that I can back and relate to my synthetic pathway or a degradation pathway. Once that is understood, or as a manufacturer, it's easy to control the impurities. So when a regulator comes in, is about the filter rates and other other inputs you use, like your charcoals, methods, etc. In evaluation of genotoxic impurities, and most importantly, this was actually interesting. So there are debates that happens, like though it happens in this case. Many of the understanding is like if USP has certain impurities as a part of the monograph, that are enough. But I have seen in my experience many times the FDA came back very heavily, saying that we have nothing to do with the USP. US legal standard, that's the bare minimum. But you cannot justify the quality of your product just to say that there are no limits for impurity USP. And the similar case with IP as well. We are supposed to give you in the beginning, as an industry, we are supposed to give you a complete impurity profile, the sources of impurities, of all kinds of impurities, and what kind of what those impurities in my product, how do I control, what control strategies I have in the process is must from the manufacturer, and also you should reasonably expect document, so that gives you a kind of confidence on the product. Regions in the EU, sterile manufacturing Annex 1, Volume 4, that also has a on the control strategies, contamination strategies, product quality system, and the quality management system has to be imbibed into these control strategies. Otherwise, it's very difficult to control. And there are many questions about the threshold, identification threshold, and qualification thresholds are not matching with the dose. So there are certain limits given, assuming that your dose is less than or more than 2 grams, with a maximum body of 65 kg, something like that. But it is the industry and regulators' responsibility to see the justification for these impurities with respect to the region, with respect to the species, with respect to the race, daily dose or a maximum or a kind of permitted to these impurities. Industry, of course, having all this experience with various regulators, industries very aware of all these, and they also getting the latest technologies that were actually available to the industry in characterization, identification of the chemical species, control contaminants, the physical contaminants, or a kind of particular talk about, and a microbial contamination was and the best available the technologies are available in the market of the best. But again, very cautious as the generic products are very driven by the cost, very much driven by the availability, very much driven by the demand. So they are very cautious, of course, and they have a set of specifications for the different regions. So I've seen companies producing for market versus regulatory market. They have entirely different processes, different set of equipment, set of strategies. But end of the day, the patient is patient. So we need to understand and the safety of the product we are administering to our friends and the fellow citizens. So the regional specs are actually creating a havoc, especially in the industry's approach, because for them it is a repetition of everything. If they have to develop 3 different processes for 3 different regions, for them generating the more data is actually cost inclusive, so it attracts some cost. Since we are working on the generics, is a primary trigger for any product that should come into the market. So having, or the industry is having, the regional specs, different processes for different regions, and different kind of strategies, marketing strategies for these product. For countries like the India and other rest of the world, we are mostly driven by the compendial. Agree with me, all regulators are here, are largely driven by specs. And though there was some thrust, some focus on understanding the overall impurity profiling of the product throughout the shelf life, I think there's a lot more attention to understand the overall safety of the product. The industry is very much geared up to absorb the technologies like the QBD and design of experiment by design of experiments, robust analytical technologies for identification and characterization of these impurities, and also the comparability studies with the reference listed drugs or approved drugs, so that we can effectively prove our generic is comparable to drug. There are a lot of advancement in the process engineering, automated operational controls, so that any adverse things that can happen to the product during the processing, either it is a time, or it is the process cycle time, or any other that can happen to the product, can be effectively controlled by good process engineering practices. That also very much evolved, especially in the majority of the Indian API companies. Formulation, I think, is pretty much evolved now. Many of the controls already exist, and there is a technological advancement in terms of robotic controls, less exposure, best practices, and less intervention of the human being during the processing. These are all available options for the industry nowadays, and that should commensurate with the risk of product what we handle at this level. And also aware that what kind of strategies they need to imbibe in these impurities in this product, in their product, especially on the key starting materials, process, and the primary packaging materials, including the product logistics management, right? Once it is out of the gate, and where it goes, how it goes, what kind of controls you need, validation and the qualification of your partners must be in place. Also qualify our logistic partners, or transport partners, our warehousing partners, and the distribution partners, how the product and what extreme conditions that are potential during these activities. And finally, and the industry is also aware, and of course the regulators also of this. We also aware of the extractables and leachables, especially in the products, and compatibility study with the primary packaging in all other drug types. Safety studies of any impurities that are beyond the safe level that is already published in the literature. So there are techniques that are available, in terms of these extreme studies for the compatibility of the primary packaging material with the drug product. The good thing is now the regulators were actually pushing the industry for testing these executing these tests for a near expiry, because sometimes we used to do this in the beginning of the maybe a third month till sample we used to expose and get this study. But there is now a commitment that is being asked by the regulators to all include this test at the last station of the stability study. So the takeaway from this, probably, I hope I'm not exceeding the time that I've given to me.
That's okay. Yeah, go ahead.
Okay. So the takeaway is of room for the advancement in the control of these impurities at industry level and at the regulators level. As Indian citizen, of course, we really need to gear up for control of which could be a detrimental to the patient safety at an. We look into this is again, the science is very clear. The technologies are available, and only thing is we need to put it in a controls at each different step. Ask for the impurity profiling and control of impurities at various steps. How do starting material, during processing, after processing, storage period, testing, and that would really help to take this subject to the next level. And certainly there is a requirement for harmonized practices, regulatory expectations, and that will of time and also ease out the problems that we have as an industry. Of these impurities is also most important when between especially the drug regulators, application reviewers, to understand the process and the potential impurities, how they are being and whether justification for those impurity levels are really acceptable. So not real specs, that is enough. And beyond chemistry, the dynamics or impurities are largely dependent on the analytical methods. So if you don't have a good analytical method, and if you don't have a data that shows up the method is really robust, sensitive, and accurate, or reproducible, then there is no meaning of the data in the drug product. So that is more important to understand first, because that's the only technique by which you derive the actual content of that impurity. That's the reason I would always believe that the process development and the manufacturing go hand in hand with the analytical research, operations, supply chain, and regulatory affairs within the industry. Process analytical development, and any changes during the post-approval a period of time, we must also be carefully evaluated during your revisit. As a regulator, as an industry, definitely we do this because any change in the process, we will evaluate internally for its impact. And we assess that product, whether it is impact is negligible or significant. If it is significant, what kind of remediation mechanisms we have or a mitigation. I think regulators also have to focus on those kind of a minor moderate changes over a period of time could really be challenging of what is accepted and approved as a part of the product approval. And need a validated processes, methods, and vendor controls beyond the release. Impurity profiling is believed to be tested, is controlled by testing, not just by testing. It has to be controlled to the roots. It should be imbibed with the quality principles. So by design, they should be able to produce the quality product, just testing. And due consideration and evidence database, due consideration the evidence and the database justifications for the impurities or the yield specifications or a control limits must be very, very important. And to my industry colleagues and also the regulators that you have to have a staggered specification levels for your drug product. For example, I might be having an impurity limit for my shelf life. Not necessarily the same I should apply as a release specifications. Probably for release specification, item specification, or for a stability specification, I can be different. At the end of the day, I am liable or responsible for, as a manufacturer, the responsible for the regulatory specifications that is approved, in which the product is expected to be during its shelf life. And periodic assessment and corrections are very important. Otherwise, all these will go because there could be a change in input, there could be a change in the chemical source, in the equipment train, there could be a change in the MOC of ancillaries you use, which may actually leach out to the product and get your product. Largely, this is what I have for the day. Hope this is not boring, and I thank you very much personally for all your patience in the last half hour. Any specific questions or anything which we can discuss further in this forum, I welcome it. Thanks for your attention, and I really appreciate it.
Thank you, Dr. Satya, for your valuable the information presented, covering all aspects of impurity from generation, source, their degradation studies, what are the expectations, then the threshold limits, analytical methods, genotoxic, mutagenic, or carcinogenic impurities, and what are the experiences you faced with regulators across the world, like their review, the comments and questions, and how the industry is approaching handling these questions and answering the aspects of the impurity profiling and about the maximum daily dose and permissible daily exposure. So all these are important topics you have covered in the time given, because each of them itself is a big topic, and it goes on forever.
Yeah, exactly.
So to put them in a nutshell, it's a very difficult task, and you did it very efficiently, and we appreciate your effort and time for all our drug control officers in enlightening us for understanding the various aspects of impurity generation, the control and the methods available, the technologies. So all sorts of things you have touched it. So on behalf of our Regulatory Officers Association, we are very much thankful to you for your time. And I have, to initiate the discussion, I'll ask you a question about the recent, the bonding topic about nitrosamine impurities, nitrosamines. So can you just talk briefly on nitrosamines for the benefit of our
Sure,
officers?
I think that's actually a buzz topic in the industry nowadays. Post this issue that is detected with the valsartan in US. So especially these impurities, to be honest, they are not new. The ages, they are part of our food. They are part of the water. They are part of any other as a part of our livelihood. So, but now why this came up, and there are a lot of studies that are happening in the last 3-4 years. right from the threat notification from US in terms of the nitrosamine impurities till the removing them from essential list, drugs list in India. There is a lot of things that are happened. These impurities are essentially any nitroso compounds in the product that can have a reaction and they generate the NDMA impurities and all other. There are 7 categories of impurities, and there are a lot of literature available now. Those impurities they are potential in almost all the drug products, not in the drug substances, not just sartans. Sartans in the sense either it is valsartan, sartan products, which are kind of antihypertensive products. Now, these impurities can be carried through your inputs, your chemical or any other industry chemicals. It can be transferred through your the water. It can be through any other aids you use or an ancillary aids you use, which can generate these 7 kinds of impurities that are having a different level, the tolerance level as a daily intake in terms of that. So unfortunately now, I think there is a pretty much advanced technologies or the methods that are published by US FDA itself. They have studied these, they have published the analytical methods by which the industry has to see the levels of these impurities in their own drug product and certify whether it is the level 1 or a level 2. For example, if there is an initial assessment with these methods is proven to be your product is not prone to these kind of impurity, a certification would be enough at this point of time. But if there's any alert, if there's this, the specification that is expected to believe, supposed to test these products to the limits that are already published in those guidelines. And we must hold those batches until we get the approval. So I think very much advancement that has happened in recent past about this nitrosamine impurities.
Yeah, thank you very much, Dr. Satya. Yeah, it's a wonderful information. I have not seen any questions on this. I think all our officers are very much carried away by the amount of information you have given. I think they're very much have a lot of things for pondering over. And one of the gentlemen has asked for a copy of the presentation. I think we can make it available online through the IDCOC website or IDCOC sources. So that's not a problem. So any other questions if anybody has?
Sir, can I ask one question?
Yes, please, sir.
Sir, in the lecture you have said that characterizations and detection of impurities in respect of our all monographs, including US monograph and IPDP and things like that. But do you think that our monograph is sufficient to characterize all these impurities to find particularly the carcinogenic material?
To be honest, to be honest, no. The monograph is a kind of a limit or sometimes a method. For example, a method could be again it varies. Some monographs, the age-old monographs will have a old techniques like TLC or by or determination by some kind of spectroscopic methods. So to precisely answer your question, beyond we need to think beyond the monograph. The monograph is actually supplied the commission, Pharmacopoeia Commission, long back, commensurating the knowledge we have at that time. But now the industry has gone way ahead, and then the process analytical technologies that are available now, especially in terms of the impurities, it's more to do with the method, because the method must to detect the impurity and quantitate that impurity at the specification level. For example, if the impurity limit is, let's assume 0.1% in the monograph impurity, for example, the method that is given in the monograph must be able to quantify precision and accuracy at 0.1% at minimum. As limit of LOQ precision, technically, a limit of quantitation, that should be 30% of your spec if you talk about the science. If it is more than that, there is every possibility that we will land up into the as a part of method variation. So the sensitivity of method in terms of detection, in terms of quantitation, in terms of reproducibility or a repeatability is a method dependent. The most of the methods we use nowadays is HPLC, but some impurities is just possible by HPLC. Probably you may need a hyphenated technique like HPLC coupled with mass spec, or you may need like a kind of methods to see whether the method is really effective. So this is into the industry until unless you get a convincing answer that how these hope that answers your question.
Okay, thank you.
Yeah.
So what to Raju sir?
Yeah, Dr. Jayanta. So I want to add a few more lines to what Dr. Satya said about
Exactly, sir.
the monograph is sufficient or not. See, there are certain things. Monographs are developed based on experience of the industry on a particular route of synthesis. So each API can be synthesized in more than one way. So different companies adopt different ways. So they use different starting materials. They may use different reactants, different catalysts. So all these contribute to the final profile of the product. So like the API. So impurity profile is different for each method of synthesis. So based on the API manufacturer's information, their knowledge, the impurity testing has to be developed by the manufacturer. So monograph is a general guidance, but this for specific testing part, we have to go with the API manufacturer, their knowledge, their information, their scientific data. So that is beyond the monograph.
Just to add on to it, sir, I'll give you a classic example so that you'll understand this better. For example, a product called atorvastatin sodium, calcium. Hello?
Yeah, right.
So, for example, atorvastatin product, there are predominantly 32 kinds of manufacturing processes that are available already published in the literature as a part of their own patents. Use the product of atorvastatin by 32 different ways with 32 inputs. So the impurity profile, what is given in the IP or BP or USP, is based on the innovator or somebody has the Pharmacopoeia Commission, based on the process they follow it. Any input as any reaction with that is associated with the alcohol, for example, I can use either methanol or ethanol, right? Methyl alcohol or ethyl alcohol. The initial monograph owner, whoever is our contributor, might have used for ethanol as a process solvent, right? Because of which they may get a diethyl impurity in their product. But the other manufacturers may use ethanol as a solvent, so that is for them it is easy to adopt. Then the impurity will not be diethyl; it will be dimethyl. So if I look for diethyl, there is no sense, right? I will be meeting the impurity profile of your monograph or IP. But the other impurity which I'm accounted is dimethyl, which is not accounted in monograph. So I must look at minimum of a monograph, any other possible impurities that will come or arise during my process, depending on the process which I follow. Hope this clears your doubt.
Thank you, sir. Thank you. Great. Sir, Dr. Rajesh, sir, please conclude the session.
Yeah. So this is, the most important, the takeaway from this today's session is about, think beyond the monograph. That's the most important point in understanding, all. And think beyond the monograph and think beyond finished product testing. That's it. So it doesn't help us if a finished product passes with the set specification. So, for example, I'll give one important aspect of, the impurity control in excipients, especially in most of the liquid formulations, we see manufacturers using propylene glycol. So
Yes.
propylene glycol is very, very, prone for contamination of diethylene glycol and monoethylene glycol, which are very, very toxic. So if those are not controlled, these DEZ and MEZs are not controlled in the propylene glycol itself, the product could be, dangerous for the consumer. So thank you very much for, enlightening us, Dr. Satya, for your knowledge, experience, and it immensely helped us. All our drug control officers will immensely benefit from your talk, and we keep in touch with you for more information and updates. And
Sure,
so
My
thank
pleasure.
you very much for your time. And Dr. Jayanta, yeah, please
Yes.
close
Yes,
from
sir.
take on this and
Yes.
close.
So on behalf of All India Drug Control Officers Confederation, I once again I express my sincere gratitude to both our chairperson of the today's session and speaker, Sir Dr. V. Raju and Dr. Satya Maturi, for sparing their most valuable times with us, and we learned a lot from this complex topic. Actually, regulators are very much benefited about the discussion. It's a very lucid and interesting, insightful presentation of the Dr. Satya Maturi. So my sincere thanks also goes to Central Council members and Executive Council members and all regulatory friends, center and state regulators and industry and academia, once and all the participants, and thanks one and all. Thank you. Thank you, sir.
Thank you. Thank you so much,
Have a great day.
Jayanta ji, Dr. Raju sir, and all other the regulators or the inspectors who are in the forum. I really appreciate and thankful to the opportunity that is given. And I'm always, as always, I'm happy to associate with you for any other things. Thank you very much. And
Sir, we are expecting much more session from you.
Thank you very much and have a good day. Yeah.
Thank you.
Yeah, right.
Sir, good night to you, Dr.
Yeah, good day. Thank you. Bye.
Thank you. Bye-bye.